Hello, everybody, and a very warm welcome to today's webinar on our Alternatives and Simulation in Education series. My name is Kathrin Herrmann, and I'm the Education Director at the Center for Alternatives to Animal Testing, CAAT, at Johns Hopkins University. And it's my pleasure to welcome you on behalf of CAAT and together with our partners from Ombion and Interniche. And so now it's my pleasure to introduce today's speaker, Associate Professor Brett Lidbury. And Brett Lidbury is a professor at the Australian National University, ANU, and he completed his PhD at the John Curtin School of Medical Research at ANU in viral engineering and cytokine biology. And he's a fellow of the Faculty of Science of the Royal College of Pathologists of Australasia. And his work spans molecular virology, laboratory medicine, and importantly for today's discussion, the transition from animal-based models towards human-relevant, animal-free biomedical research. We are very much looking forward to his talk. Brett, thanks for being with us, and the floor is yours.
Thank you very much, Kathrin, and thank you everybody for attending, and lovely to see so many people. I can see here 92 participants, including the panel. A great big thank you to the Centre for Alternatives to Animal Testing, CAAT, of course, Interniche and Ombion. And let me say how delighted I am to see something like, particularly with Ombion, and of course others, interested in animal alternatives in the biomedical sciences and biomedical research. I've attended several World Congresses, and for the first few I felt very lonely talking about biomedicine, but that's clearly changing. My name's Brett Lidbury, and I'm based at the National Centre for Epidemiology and Population Health at the Australian National University in Canberra, Australia. Quite a strange departmental pairing for someone like myself whose career has been in the biomedical sciences, but that's a topic for another seminar at another time. So, yeah, from in vivo to in silico, the transition to animal-free fundamental biomedical research. Now, I'm in the habit nowadays of, rather than putting thank you slides at the end, I do them at the beginning, so I can give them the full attention they deserve and not rush. It wasn't easy. And there was an amount of serendipity and synchronicity that really helped me move from the fields. While biomedical science was still my focus, the moving focus away from animal models, of course, was significant. And I have slides. I have a number of slides to share tonight, but it was almost, when reflecting on how to present tonight, it was almost a talk that I could give without any visuals at all, without any slides, because it's really a story. And I'll talk more about the story in a moment, about the good luck and good fortune that came my way. But one thing that Kathrin's pointed out, she's quite correct, and we're all aware of it in Australia, is we don't really have like a focus or an identity in Australia around the 3 R's, NAMs, or animal-free research toxicology. But in saying that, there is an enormous interest in this country. We just don't have a label for it at a national level, like in Europe and in North America so much. So very early in my time, I started back at the Australian National University. I'm a graduate of the ANU and was away and came back on a fellowship, and the fellowship was targeted specifically at developing animal replacement alternatives, and absolute alternatives, not partial alternatives, but really not just doing my own research in the area and innovating in alternatives, but also promoting it amongst my colleagues, students, and the wider research community. And I was very fortunate to have early on the Director of Research Integrity at the ANU, Simon Bain, and assisted by Kelly De Bono and the Animal Ethics Committee. And there was also quite a push at the University of Sydney. Malcolm France and George Grau were instrumental, and I was quite involved in their program for some time in their research education about actually educating graduate, postgraduate and honours students in how to replace animals or how to think about alternatives. Now, very early on, I was quite delighted to be assisted by the lovely people, CAAT Europe and CAAT, of course, USA, so the CAAT network in Europe and the United States, Lena, Marcel, Thomas, Helena, who I know has moved on, but Francois as well. Spent a lot of time with Francois up in Brussels, which was lovely. And also early on, when I was just sort of flashing around the place, a little bit lost in Australia, I had significant support too from what became the Circle Group at the Radboud University in Nijmegen in the Netherlands, particularly Merrill and the rest of the team there, the Circle team. And the ongoing support, I'll just go through this, continues at the ANU. I'm an advisor to the Animal Ethics Committee at the ANU on alternatives and replacement and the 3Rs in general. But also, I saw Daniela there. It's really good that I can also thank the Transitieproject Free Innovatie Network in the Netherlands, which I haven't done a lot with recently, but they I joined that network and have done some work together and really value that. And within Australia, we're very lucky to have the Animal Free Science Advocacy Group, formerly the Humane Research Australia, Helen, Rachel, Natalie. From there, need a big shout out. And through all of this, right, really almost from day dot, my friend and colleague Alice Richardson, she's the Professor of Statistics at the ANU, and Meryl Ritzhoitinger, I mentioned the Circle Group on the previous slide, but Meryl's been a wonderful support, really, almost from day one of this initiative. And in fact, I've spent several months here and there with her group when she was based at Radboud in Nijmegen, and there to learn how to do systematic review. So thank you to all those people. I don't want to say for a moment that it was all my doing and I just waved the magic wand and it all happened. I needed to garner support, particularly from Europe and the United States. As I mentioned a moment ago, it's almost a talk that I could give without a slide, but I thought being a educational environment that would not do. But there's a real transition, and I mean, I'm no expert in the archaeology or anthropology disciplines, but really, when you think of any of our careers, there's a tribalism. And I became fully sort of inducted into a way of thinking very early in my education as a scientist. And my undergraduate education at the University of Newcastle here in Australia was in the biological sciences, and I was very fortunate to be in a program that worked from molecules right through to ecology and environment. And so there's a real focus, of course, on animals and plants and what all that means. But through that period, and of course into my later studies within the environments that I worked in within Australian and other laboratories here and there was a genuine belief that a mouse was a miniature human, that the mouse actually fully represented human physiology and pathology, which of course, just on a moment's reflection, is insane. Phylogenetically, rodents and primates diverged something like 65, 85 million years ago. They do possess some similarities, of course, which have been useful. Other mammals, of course, have been useful to understand. But in the more modern contemporary context, it's almost irrelevant. And I don't need to explain why to this audience. Now, as a younger man, as a young scientist, I was completely inured to the culture and identity of being a scientist and biologist. And the doctoral experience at the John Curtin School of Medical Research at the ANU really even more strongly embedded that into my way of thinking and into my own philosophy of doing science. And being a medical researcher, which by definition is research into human maladies, into human disease and health. I've just underlined on this slide, if I can use a pointer, this MHC fame, which I'll talk more about in a couple of slides. So this is just, this is my very first paper that came out of my research year, what we call honours. And it was to do with understanding how basically embryos can be embedded in a mammalian system and not be rejected because the fetus is what's called a semi-allograft. So we use murine models, and I just highlight down here the animal's BALB-C and CBAAH. You can see here different MHC haplotypes were employed in these studies. So it was very much an animal focus from very early time in my research career. And this paper here, published in the Journal of Virology in 2011, so that's 2011. That was the last paper that I published using any animal model, in this case, again, a mouse model. And there was a paper 3 years later on some work that I contributed to in the 90s, but in terms of driving the research in mouse models, this was my final one in 2011. See here, the mouse model, which, basically it was work that I led on characterising variants of a virus called Ross River, which causes significant disease in Australia and is related to chikungunya virus, which I know sadly Europe knows about, mosquito-borne virus. So we used the mouse model there, but that was basically it. So that was in 2011. And in 2011, I was transitioning away from animal-based fundamental biomedical research to animal-free, which I'll talk more about in a moment. Now, I mentioned 2 slides ago about my early influences. The culture as a doctoral student was immensely strong in the view that animals were valuable for understanding human physiology and pathology. And that was like an attitude you can find in other places, but it was particularly strong in this institution, the John Curtin School of Medical Research, because Nobel Prizes had been awarded to researchers from that institution. One of the most, or there's 2 particularly famous cases, but this one was very, very fundamental. It was the understanding of what's called MHC restriction. MHC, major histocompatibility complex, or transplantation antigens. So you know about if you graft between people, even brothers and sisters, unless you're an identical twin, you can't easily graft tissue between humans because of the MHC complexes. But they were discovered in the 1970s by John Curtin researchers to also have a role in identifying virally infected cells, and really basically explains what's called cell-mediated immunity, T-cell driven, killer T-cell driven immunity through this process. And this, for Doherty and Zinkernagel, won the Nobel Prize in Physiology or Medicine in, from memory, 1996. So the other one, don't have a slide for it, but earlier than that, Sir John Eccles won the Nobel Prize for Physiology or Medicine from the John Curtin School of Medical Research for his work using squid. So he was a neuroscientist, and squid axon was very easy to manipulate in the lab, and he discovered the electrical sort of foundation of nerve signaling, and that attracted a great accolade as well. So there was that very, very strong culture. So this slide that I'm showing now, you should see a black mouse and a white mouse. Basically, my advanced studies, my research studies as a student, were interested in immunology and virology, particularly what you'd call virus-host interaction. But basically, to summarise it, how do viruses make you sick? So this slide summarises many years of the work, and for those of you that have worked in the lab, you'll see here, this is an electron micrograph, the green blob here, of viruses, graph of viruses. This is some muscle histology. We measure the viruses in various tissues and in blood, and we also look at the expression of genes. We did a lot of gene expression work to look at what immune or inflammatory genes were switched on or off under certain conditions. Now, the thinking here, it's the black mouse and the white mouse that's important. And of course, nowadays you'd have a genetically modified mouse as well, probably. But they had the black mouse, the C57 black 6 mouse, has a very strong what's called a Th1, or a delayed type hypersensitivity, or sort of cell-mediated driven immune response. Whereas the white mouse over here, the BALB-C, has a very strong antibody, or a Th2, a T helper 2 driven response. So they have fundamentally different immune sort of angles, if you like, to help untangle how we how we understand viral illness and pathology and susceptibility to infection. So that was the world that I come from, and I'm not going to dwell much more on it, just to say that's a lot of my work. So for context, let me see, to explain this, I was fully embedded in the mouse experimentation world, at least till about 2007. By 2011, I was very fortunate to have earned a fellowship, I mentioned before, to return to the Australian National University and to start a program which was focused entirely on animal-free and absolute replacement, so replacing animals entirely from fundamental biomedical research, which of course now, and it's so lovely today, we talk about NAMs. And what I do when I talk to people is talk about also it's human-focused research. We're not spanning the vast species barrier like we do with rodents and primates like ourselves. So the story behind it isn't just, and I was saying to Kathrin this morning when we spoke about the talk, that I could present it as just one of those epiphany moments where you wake up one day and go, I'm sick of this, I need to change direction. It's like you have, a voice from heaven talks to you and say you need to change direction. And people, I'm sure, can have those experiences. But for me, it was, there were a combination of factors, including in my private life, had young children, family situation wasn't going well, I had care for children, and other difficulties in the background, and I just simply couldn't keep up with the demands of running a laboratory. As you know, it's very, very competitive to run research laboratories. So I was in the wilderness for 2 or 3 years and gradually turned to sort of other ways of thinking. So I can't ever claim that I had a particular insight that led me to this realization. It was a very gradual process that was encouraged by a number of events, both within my academic professional setting, but also within my family life. But behind all of that, I've always believed, although the system nowadays doesn't quite encourage it, I believe, but I still very, very strongly believe, as an early influence, that scientists must innovate. They must lead innovation. And I'm afraid, and I'm sure it's not only a problem in Australia, is that the difficulties with funding and other challenges don't encourage innovation. So on top of all these other factors that came into play to help me, there was also significant luck to provide resources and an opportunity to move. So I want to say from the outset, it wasn't just a sort of come to God sort of realization moment. It was a situation that developed, and I was just in a good place. Once the opportunity arose, I was in a good place to take it. And I've never, ever regretted it. So that's how I came to it. So for those of you who may be involved in animal-focused research on rodent models or any other non-human sort of model for fundamental research, it's not necessarily straightforward, but what I'd say to you is it won't happen immediately, but just be true to your convictions and the opportunities will arise. And in fact, there'll be more opportunities now, I'm sure, through this process, because the world is waking up slowly but surely. So I was just very keen to emphasize that. So here I am, and before taking the formal position as an associate professor devoted to alternatives to animals, I had started working on some ideas outside of the laboratory, which were very helpful. I have some slides to explain in a moment. But where do you start? And a good place to start in this case was a paper that's now over 30 years old, but of course Michael Ball's published something in '94 about, well, how do you replace animal procedures? What are alternatives? What do they look like? What's possible? And I read this paper, and this is just a quick summary of things that Michael suggested: data storage and sharing, physical chemical modelling, etc., mathematical computer models. Human studies down the bottom, which were quite useful for me. So, yeah, lower organism, how you define that. Of course, we'll leave that for another occasion, questions of sentience. So all of those sorts of things sort of helped give me some direction. But also, as I mentioned very early, there were other people in the early phases who were very supportive and very helpful in terms of discussions and support. So what I found, and I continually find this a little bit annoying, but in fact, even last Friday, I caught up with an old colleague who's also a science academic, and they're quite, at times, quite disdainful, some of these colleagues, of philosophy. And I really disagree with that disdain. In fact, I applaud philosophy. And as I say here, one must become philosophical. And the evidence for that is I was very fortunate to, again, quite early in my time in this field, to have been invited to attend workshops and other events up in Sydney via the philosophy department at Macquarie University, which is based in Sydney. And long story short, ended up publishing or co-authoring a paper with Cynthia Townley, a philosopher at Macquarie, called Mousetraps and How to Avoid Them: The Convergence of Utilitarian and Scientific Cases for Limiting the Mouse Model in Biomedical Research. That was published in 2012. And this whole business with talking to philosophers not only led to a nice publication, but also encouraged me to delve into that literature and think in a different way about how one can meaningfully move and find ways to replace, but also develop that philosophical basis. So what you're dealing with, and I'm embarrassed to admit this, but up until this point in my career, so we're talking 2011, 2012, I'd never thought about issues like external and internal validity. I certainly had never heard of things called hams and cams, which I'll describe in a moment. But that very issue, which seems so obvious now, about validity. Of course, a mouse model has tremendous internal validity. You repeat it in other mice, but the translation to other species, and of course in the medical research case, translation to humans, you also need external validity. And philosophers talk about this stuff. And of course, philosophers write for philosophers, not scientists. And sadly, a lot of this literature isn't really appreciated in the area or within the disciplines that really need to understand it. So that was part of the journey. And I encourage everybody else thinking about this to become philosophical. Now, there's a little bit here, but another, in this context of interacting with philosophers, thinking in a more, I was going to say lateral, but a wider disciplinary context. So rather than just focusing within what you do, look sideways. So broaden the focus, talk to colleagues outside. So this was a very long paper published in 1995 by La Follette and Shanks in the Philosophical Quarterly. And this is just a very small summary, but it really jumped out at me. Now, they talk in this paper about many things, but what really resonated with me was this concept of a ham and a cam. A ham, H-A-M-S, is a hypothetical analogical model. A cam, C-A-M, is a causal analogical model. And through reading this paper, it basically explained to me what you're trying to do as a scientist. And that is largely what we're doing is testing hypotheses. And where we become confused is to understand that while we're testing hypotheses, we're not necessarily identifying causes. So the system I grew up within was that a mouse is a causal analogical model. That is, we can divine from a mouse or a rat causes of human disease. Whereas in fact, they're simply a ham, a hypothetical analogical model. Ultimately, the results from your ham, whether it be a mouse model or something else, ultimately needs to be tested in the target species, which is a human. But the key thing here is, and I'll read this quote from the paper, of course, if animal models can serve as hams to spur research, perhaps clinical investigations, cell cultures, computer simulations, or epidemiological studies might be effective hams as well. Ham, hypothetical analogical model. At the very least, in yellow highlights, these other methods need no longer be construed as poor cousins to animal research. They may all become more important part of basic biomedical research. This was a moment that really made me sit up and take notice to really understand what one is trying to do as a biomedical researcher. So again, I could have included hundreds of slides tonight, and I must say I did struggle somewhat deciding ultimately which 20 slides to include. So now this isn't new. This system that you see in front of you now, the in silico pattern recognition system, with lots of arrows pointing everywhere, was something that Alice and I published in the proceedings from World Congress number 8 in Montreal in 2011. So this was in this phase of transitioning, and again, I'd been appointed to a fellowship. I had this opportunity, talking to friends, colleagues, philosophers, everybody. This is what I put forward as my hypothetical analogical model, my ham, to replace the animal. So it's a combination of in silico pattern, or what I was calling at the time, pattern recognition, so machine learning. So using algorithms rather than the black and the white mouse. So you can just sort of see here, there's a collection, compilation and cleaning of human data. So of course, by very nature of it, it's human data. And for these types of studies, we used data collected via hospital pathology departments, which of course, as you may realise, represent a lot of human biochemistry and physiology. So given a particular disease of interest, you can actually use algorithms to, rather than use or find a white or a black mouse, well actually you can find a white or a black mouse, but within all that human diversity. So the whole idea is to use algorithms to take very complex human data and then separate it into sub-cohorts that represent some aspect of infection, disease, inflammation, whatever it is that you're interested in as a medical problem. So the system that I propose is you have the retrospective data modelling phase to look for patterns, and then a validation phase, again, pathology blood tests are at the centre of this, and then follow the arrows up to then, looking at other markers, other in vitro studies, gene or protein expression, those sorts of things. And one area where I received some criticism very early was to say, well, how do you examine mechanism? Yeast culture, there's other ways, but here's another updated slide. Mechanism, human on a chip, of course, nowadays. We now have human or organ on a chip technology, 3D cell culture, yeast culture, but also I mentioned, of course, my connection with Merrill and the Circle team, systematic review, which is something I've been very interested in to help actually unpack, I suppose, some of the findings around the early machine learning pattern recognition phases, to then turn to a systematic review to help uncover potential mechanisms around that. So that's basically what you see now is, in big blue letters, animal replacement alternative system. That's my mouse, but it's even better because it's based on humans, human data, but also human volunteers. I have been involved in many studies where we've recruited human participants into research and, of course, conducted studies with their consent. So what are these algorithms, etc.? This is one I'll build up. This is quite an early one. We did on understanding chlamydia, so chlamydia infections. We had a project with the Canberra hospital. They were very interested to know about a spike in a respiratory infection caused by chlamydia. And you might recognise here, I'll just flick through fairly quickly. There's the percentages I'll explain in a moment. So we had 451 patients. In the end, you come up with a bunch of, or a collection of decision rules, which improve accuracy as you go through by adding certain parameters. One here is age, of course, can be important, but platelet count, white cell count, and markers of anemia like hemoglobin and red cell distribution width. So putting these things together and determining the thresholds, whether less than or greater than a particular concentration of platelets or an age or whatever it might be, you can actually form up some decision rules and help understand what's able to help you, understand who's sick and who's not, for example. We did some work too. I did some work with a genetics colleague who was interested in Alzheimer's, and it's a similar idea. You can see here. I can't remember all the symbols here within the boxes, but it's the same concept. Is that you have basically data derived from brain chemistry or brain imaging, which could actually tell you something about the brain chemistry, and you can then just bifurcate based on decision rules and actually end up with yes and no status, illness, health, whatever it might be, or levels of particular chemical markers within the brain. And this is done on humans. We don't need to take a mouse or a rat and extract its brain and then do histology or any other sort of laboratory analysis. We can actually do this in a living human being. So the results are relevant immediately. So these types of analyses in the language of machine learning, it's called recursive partitioning. Better known in the more straightforward language as this is an example of a decision tree, and that's a decision tree. And there's also things called random forests, where you can actually run lots and lots and lots of decision trees to look at the most powerful predictors of a particular outcome, whether it be Alzheimer's, Parkinson's. We've done a lot of work using these methods to understand hepatitis B infection, so hepatitis B virus. There's another one that we really like called a support vector machine. This is a very, very powerful algorithm that can separate, so you have the red dots and the green dots. And what you have through the algorithm, this is just a simple linear kernel. It sets down what I call through the support vectors. They put in this sort of wedge or this sort of boundary, which helps separate. So up here, the green dots, you can think of that as the bulb C or the white mouse. And down here in the red dot, you can think of the C57 black 6 or the black mouse, for example. So you can see earlier from my earlier slides that I was trying to understand susceptibility and responses to viral infections through different types of mice, different inbred strains. And I'm not saying this is as clean as an inbred mouse, but at least we can separate populations, diverse human populations with a lot of variability. We can have some success in modelling those data. And this is an example, again, no need to know the details, but you can see the blue and the pink. And this is a nice one to demonstrate the power of the support vector machine. And again, human data results are used as a foundation. And we have some measures here, age, whoops, go backwards, age, platelets, and I think globulin level to predict hepatitis B surface antigen. So again, think of the pink as the black mouse and the blue as the white mouse. And we change parameters and you can see the pink changes and joins up and you have two separate categories of blue here and you have different dynamics. So it's not just a static type of modelling outcome that you might be used to with correlation regression, some of the usual inferential stats, but it's a more dynamic way of actually looking at variation within, complex organisms like a human being. I'm not sort of saying it's absolutely perfect, but it has given us some success. And thanks to mathematicians, statisticians, and computer scientists for basically developing these incredible algorithms to help us unpack and I suppose, pull apart, unbundle very complex data and results based on human biology. Now, Kathrin asked me, and I'm so pleased, I'm coming towards the end of the talk. I'm very keen to generate some discussion, but thanks to Animal Free Science Advocacy for this slide. It has a very non-flattering title. Australia is falling behind the rest of the world. I was saying to Kathrin this morning that I really, as an Australian, I really don't like it. I mean, often in the media, Australia is lagging behind in this or that or something else. Now, nowhere's perfect. We're very good at some things and not so good, just like any country. But there is a point here, though. We are falling behind. But what I'd say about this, and Rachel and the team at AFSA have pointed out amazing things happening in the Netherlands, and that's why I go there so often. I love going to the Netherlands. It's so dynamic. I gave it the Dutch name before, but they point out here the Transition Program for Innovation Without the Use of Animals, the TPI program. And I know there's a lot of spin-offs and startups and all the rest of it going on around this very question of animals in research. The US is, in spite of everything going on over there, have done some incredible things with the FDA Modernisation Act, and of course last year announced that the NIH won't consider fundamental research studies that use only animals, or mice particularly. They must have a human element. Things are really shifting. And of course in the UK and the EU, there's much going on there as well. I'm not as aware of it, but there's huge things happening. And the EU's, for me, always been a source of inspiration as well. So lots going on. What I'd say to this unflattering headline is to say, yes, we are falling behind. So we just don't have an entity that we can put a badge on, like CAAT or the NC3Rs or Circle or Zebit or whatever it could be. We don't have that, but we do have a lot of energy in the medical and the biomedical research community, and people in this country are doing quite extraordinary research without animals, but it's not called a NAM, it's not called an alternative. So there has been, as far as I'm aware, discussion in Australia back into the late 1980s about actually developing strength in this area, but it hasn't been grasped. That opportunity just has passed and it hasn't really developed. In fact, the Australian Senate, the Federal Senate, did have a discussion in 1988 about this very question, about we should set up an institution that's devoted to research without relying upon animal models. But that opportunity passed and it hasn't really, well, I'll save it. It has returned, but it's taken a long time. So, yeah, we are falling behind in that regard. But what I'd like to say to my colleagues in Europe and anywhere else who's listening tonight is there is a lot of energy around this and we are starting to catch up. And just to finish my formal presentation, moving on from this slide is, and great credit to the government of New South Wales. For those that aren't that familiar with Australia, we're a country and a continent, but we're made up of 6 states and 2 territories. And New South Wales is a state of Australia, and each state has its own government. So through the health departments in New South Wales, they've actually awarded, it's not a lot, but as a start, they've actually started a network and awarded some funding for non-animal technologies in research. So again, you can read here, there's a web address as well. A research community established by the New South Wales government focused on facilitating collaboration and advancing new technologies that do not rely on animal testing in medical research. Brilliant. A small start, but a start nonetheless, and huge credit to my colleagues in New South Wales. Now, at an even more exciting level, again, those familiar with Australia may have heard what we call here CSIRO. That's the CSIRO, the Commonwealth Scientific and Industrial Research Organisation. They have written a white paper and generated discussion. And this is official news now. The CSIRO charts new industry for non-animal models to advance new medicines. So this has been explored and is serious with the CSIRO, which is a federal entity, happy to drive it and to encourage a move towards animal-free innovation to advance new medicines without the use of animal models. So, of course, to do that, you need to develop non-animal models. And The centre from New South Wales, I think, have already, or they're in the process of establishing an organoid laboratory. And that's going ahead at pace. And there's a few of us out there looking to continue with this network. I'm a member of this network and wish to collaborate more widely within Australia, but also with our international colleagues. So that's the formal part of my presentation. I have a couple of other slides, but we can come back to them if you want. But basically, I'm finished.
Thank you very much, Brett. That was very interesting and inspiring, how you made this transition. There are some questions for you. I will just start with them. And then I also invite our co-hosts from Ombion, if they have questions, to just chip in. So I have one question. It says, "Hi, Brett, fantastic talk. You mentioned AFSA at the start of your talk and later, because that was said earlier. A great organization that I'm lucky to work with through ICAPO. How have you, as an academic, engaged with and collaborated with animal protection NGOs through your work?"
Yes, no, thank you for that question. And yeah, it really started. And I think with so many things, the COVID pandemic created some inertia. But before COVID, I was in quite regular contact with the former head of AFSA when it was called the Humane Research Australia. That was Helen Marsden, who very, very sadly is no longer with us. But Helen and I, it was so incredible. We actually live in the same state of Australia. We met for the very first time at the World Congress in Prague. And it was love at first sight. And Helen invited me to come on board as a scientific advisor. So what that meant day to day wasn't it was nothing like jumping out of a plane with a parachute or anything really exciting or adventurous, but it was just providing a lot of advice. And documents and other opportunities would come to them. So it was just really that, and helping prepare materials. I can't think of a time where I spoke for them particularly, but I was more in the background with them. But in saying that, I'd welcome a role with AFSA in any dimension. So it was really an advisory role. And some of that advice was also aimed at the people within HRA themselves, in that, and I'm sure people from other parts of the world can sympathise with this. My approach really was to say, look, when you meet someone who's involved in animal research, don't demonise them or portray them as a monster. It's an interesting thing. There's sort of two worlds. When you talk to other scientists involved in animal research, there's the person you meet over a cup of tea in the cafeteria, and there's a person you meet at the seminar or in the lab. The person in the lab or the seminar is very robustly in favour of animal research. But the person you speak with over a cup of tea says to you, I wish I could find a way out. I don't like it. I wish there was some way I could change. Can you help me? So I'd like to say, like, again, at a philosophical level, I was influential at that phase anyway, to say, well, look, when you interact with people and engage, don't look at them as monsters. Try and engage with them in human, and don't necessarily go to the extremes. We've seen examples in the UK and elsewhere. Just try and engage them more in the middle. So nothing purely exciting, but just in the background to give advice of how to talk to academics and to researchers.
Yeah, and obviously we have to be clear here that most scientists started using animals. So this is the norm, and this is what you learn at university. Is still a focus on it. And that's why we are actually doing the series here, to show that there are now alternatives and you can simulate a lot of things. So, but back when many of you started, obviously it was different. So it's you explained it, I think, very well, how you got to do it and how you changed. So another question: your replacement framework integrates systematic reviews, the human data pattern recognition, omics, and validation. And what do you see as the biggest cultural barrier for biomedical researchers in trusting machine learning derived insights as much as they trust animal data?
Yeah, thank you for that question. Yeah, it's I commonly come across, and people are right when they say that. So I or often in the past, would go to the University of Sydney, excuse me, and at the ANU and elsewhere and give a talk saying, look, the animal models, like you're a medical researcher, a biomedical researcher, your focus is to explore, investigate, and make discoveries to improve human health. Okay, medical research. And even by 2015 or thereabouts, it was pretty clear, like we talk about the 90 to 95% failure rate. Look, if you really want to help people, which I think is genuinely people's intention, you need to move out into human systems. And what they'd say, because I'd give my talk, this is what I do, and I've learnt to code and develop another skill set while retaining a knowledge of biomedicine. But they say to me, Brett, I've spent years learning how to do PCR, cell culture, animal experiments, and now you're asking me to learn all of this as well. So there is a natural barrier there. So what's my answer to that? The first thing is to say, yes, I see your point. The second thing is to say, look, again, like I did with philosophers, look sideways. There's a real issue in my mind that's still current today, and scientists, particularly in academia, on that cutting edge research, can often think in themselves they need to be the lone hero. And that's why I took so much time early to say, look, there were so many people along the way to help me. And it's the same thing to say, well, look, retain those skills, but learn a bit about machine learning and collaborate with someone that understands it better than you. So we're not asking you to change entirely your career. I've done it, doesn't mean you to, but find a colleague who can help you. Or colleagues, plural, find a team.
Yeah, yeah, definitely. And thankfully, I have to say that there's more and more opportunities now at universities to, yeah, to get training in non-animal methods.
Definitely.
Not still, obviously not as much as we would wish for, especially starting at a bachelor level, from right from the start. But I think it's improving in
I
many
agree.
places. So there's another question. Thank you so much for your talk. I'm the lab manager of a research aquarium in the UK, and I help support various undergraduate practicals. So different from replacement and research, but many principles will be shared, I hope, including physiology of shore crabs and blue mussels, trout autonomy, and reproductive biology dissections, and handling embryos from various species. We are looking to phase out all animal practicals by 2030. Academics are worried that the university will be less attractive to prospective students if some of the hands-on lab experience with animals is reduced or eliminated. Do you have any advice on how we can keep teaching standards high with hands-on experience, but limit the use of animals in teaching?
Well, yeah. So look, I really agree with that. The way I've always seen science, some of my work early on as an academic was I was also involved in education, and I set up a course, an undergraduate course subject in molecular biology. And my thinking always was you can't be a theoretical molecular biologist, it's hands-on. And I think in the context of that question, you need hands-on experience. All I could suggest is now there's a couple of things, and I don't think they're entirely the answer. But even from 2011, going to my first World Congress, I was amazed at the models people were producing made from latex and other materials, for surgical practice. And so there are companies out there, and I cannot think of the name of them. Maybe someone else can, that produce actually replicas of animals or that are basically have the same consistency, and they have the organ systems, but they're not a live animal. They're just a replica that one can work on. In fact, I was contacted by the College of Surgeons a few years ago, and many of the young surgeons coming through said, no, we don't want to operate on pigs. And I did actually find some information of a company that could actually give them a replica to practice their surgical skills. I'm sorry, I can't think of the name. Now, the other thing is, in terms of some of the laboratory work, now I don't know exactly what it is that what sort of experimental techniques and what sort of aims you have, but, and this is a philosophical argument, is where do you cross the line with species? And I know in the code in Australia, any animal with a backbone and cephalopods, so squid and octopus, they're sort of seen as higher up. We have mouse and rat, we have dogs and cats. But below that, you have C. elegans, you have fruit flies, you have Galligularia, like a particular sort of marine mammal that I'm sure you, marine mammal, sort of mammal, that's a type of worm, sorry. Yeah, what you'd call, and I don't mean this in the full sense, but a lower organism, like an invertebrate system, fruit flies, etc., C. elegans, which if, for example, you want to learn to do some molecular biology or cell culture, you could use them just as well as a mouse or a rat. So I'd encourage people to think very hard about some of those types of models of the invertebrate animals, if it's something that really needs biology, that is a living system.
Yeah, and actually, this is also what we will cover in this series. So stay tuned, because there are so many synthetic models now where you can actually do surgery on, as Brett just mentioned. For example, there's Syndiver or Dave, it's pronounced in Florida. It's a company that not only has a human model that you can do surgeries on, but also a dog model. And there are more and more such opportunities. Also, so how the series here started was actually in Rio as a pre-conference of the World Congress on alternatives and animal use back in end of August it was last year. And we had a lot of also different representatives internationally, and if there are, for example, restrictions with funding, there's a lot that you can actually do yourself, like make models yourself for the students, and it's still hands-on training, basically. So I just stay tuned to the series, and we will cover more. And we also hope to publish the videos from back in Rio from last year soon, because we had a film team, but it's still in progress. So the next question for you, Brett, is also first, the colleague says, thank you for your nice talk. What would you say needs to change on a policy level for NAMs to come into regulatory acceptance? How much of a problem is the general bias towards animal experimentation?
Yeah, there's many levels to that question, and therefore many levels of answer. At a policy level, it's a fascinating thing to see that there's, I suppose, I think it's more than lip service. I don't know if people are familiar with that. It's just, just saying it, or virtue signaling that, yeah, we care about this. But in the field, like in the labs, you don't see progress. I think, for example, as Kathrin knows, the Australian Code of Practice is being updated at the moment, and I've sent some commentary and feedback to that organisation that's convening that process. And look, this is part of my answer. The type of thing that I see as a problem is, yeah, this lip service saying that, oh, now this is important, but ultimately that not translating into practice. So the type of feedback and commentary I'm providing is to say, look, we've been dabbling for too long, and the principle, our code of practice in Australia is based on Russell and Birch's 3 Rs, as published in 1959. That's the philosophical foundation for it. But in practice, it's just too easy. And when I say too easy, I don't mean that it's always appropriate, but researchers can just say, well, there's no alternative. We need to study a whole organism, and that's it. So there isn't enough engagement from the other side. So from the policy side, there's not enough engagement saying, well, actually, come and talk to me and we'll show you some alternatives. So it's almost like there's a little bit of fear of the scientists, and yeah, it's maybe not really fear, but it's just that it's just too hard, and there just needs to be a framework in place now through the policies to say, no, it's not enough just to say, oh, we had a quick look online, we couldn't find a validated alternative, that's the end of it. But it's a way now to actually push through to say, no, in fact, there is an alternative to this process, and it is available through these means. Let us help you or explore it further. Don't just, committees shouldn't just sort of accept that anymore. But for the committees to do their role like that, they need to be supported by the policymakers. And again, it seems to be some distance. There doesn't seem to be that sort of flow of consequence or authority through from the policy to the actual laboratory. Now, the other thing, though, is I do recognise that scientists today, and it's a global issue, are having trouble finding funding. And in a way, that reinforces the need to stay in the existing animal model or whatever they've built their career on. So this needs to be, and it's an obvious thing to say, and it's boring in a way, but it's just so fundamental. If just appropriate funding. So the policy, that's fine. And now let's back it up to say to people, all right, we want you to change. We think you can do more relevant human-centered research through this human-centered method rather than a mouse or a rat model. And here's some funding to help you bridge that gap to actually there's a word I'm looking for, but to actually, change direction to actually find a new path in your research. So we'll have support. It's not just words on a page. I'll stop there. I don't know if that's quite covered it.
Yeah, actually, you kind of also touched on some other questions already that we have received. For example, was the question, despite the evidence about poor translation, why is that still a thing? And then coming back to oversight and policy, I would maybe add to what you already said that unfortunately the governments where the oversight bodies are not very well informed about NAMs, and that's a huge problem, of course, because if you're not knowledgeable about it, how are you going to go against not authorizing something? Because depending on where you're based, what country, it's really difficult for the regulatory body to not authorize something. That's how we see that most of animal research proposals actually get authorized.
That's right. Yeah, they see it as just writing words on a page is sufficient. No,
Yeah.
there needs to be action. And the word I was looking for was to help people transition. Yeah.
Yeah.
So, and that needs support at policy level, but funding level, etc. And people will do it. I'm sure of it.
Yeah, I mean, there was the question about how to improve the scrutiny before the license or the funding is even received. Do you have a few thoughts on that? How can we do that? We already said that there's not oftentimes not sufficient expertise in the committees. So what would be your ideas to improve that?
Yeah, again, as a concept, in Australia anyway, the way ethics committees are constructed, it looks fine. But again, you have human interaction and human nature combining. And in Australia, the responsibility for enforcing the code of practice comes to the ethics committees. And I suppose what we're alluding to here is that ethics committee needs to be supported by a bigger, more powerful entity outside of that. So, I do know anecdotally that people who come on to ethics committees as what are called lay people, so they're non-experts, but they're just they're community members to give voice. They often feel a little bit intimidated. So it's not about actually the personalities. It's about, a lot of it comes down to the chair of the committee and how they actually recognise the different views. And I've actually seen it in my experience that you can have some people who are animal researchers, they become very cross and a little bit, well, maybe not threatening, but it can be quite uncomfortable. And to solve that issue of intimidation or feeling in some way inadequate, again, there needs to be the support from on high whether it's from within the university or outside, to come out and actually. And I think from what I've seen, of course, I can't say much. I can't say anything about details. I have seen improvements there. Ethics committees feeling emboldened, and that often comes from the community members speaking up on the behalf of the animals. What I say in my role, and I'm not a committee member, I'm an advisor, is to keep emphasising what good science looks like. Keep reminding people to say, well, you want to have an impact in health, you want to make discoveries. This is better science. I mean, there's even a situation, and this isn't unique to Australia, where if you look at the reduction R, to encourage researchers to do power calculations or to statistically design their experiments. These are the sort of things that still aren't universally accepted and that we're still dealing with researchers in the field to give more robustness to the work they do if they must use an animal. And that's a truncated answer. There's much more I could say about that, but we're at time. But thank you for the question. And you can find me via the ANU website if you want to send me an email.
Yes, that's great. Thank you so much, Brett, for this wonderful talk and sharing your experience. And just there's one comment in the Q&A box. There's a replacement checklist in the UK designed specifically to support lay members and ethics committees to challenge review of animal research. And I will share this checklist in the chat with you.